All right, so this is my first kind of explanatory test video that I'm going to be posting. And so let's begin. So the first question is, this method of horizontal gene transfer requires the donor cell's death. And we talked about this last Monday. There are three types of horizontal gene transfer. Transformation is the first, and that one requires the genomic material to be floating around naked in solution. So in order for that to happen, the cell has to die and kind of spill out all of its genomic material all over the place. The second is transduction, which is the transfer of genetic material by a viral intermediate. And generally speaking, when leaving the donor, the virus is going to kill the cell. So the donor cell ends up dying, the virus kills it, but it takes some of its DNA along with it to a new cell, the recipient cell. So in that one, the donor cell dies as well. And the third is in conjugation. And in conjugation, if you'll recall, the cell is going to have a plasmid that codes for a pilus, and that pilus is going to allow for the transfer of the plasmid material from a donor cell to a recipient cell, and then the pilus breaks, and both cells sort of go away all on their own. And so with that one, both the donor and recipient cell survive. So which ones require the donor cell's death? That's going to be transformation and transduction. Question 2, I totally screwed up on. I meant for this answer A to be specialized transduction, not generalized transduction, and I just totally wrote down the wrong word, and I'm sorry, and we'll restore those points. Question 3, which of the following statements about conjugation is false? Both donor and recipient survive conjugation. We just talked about that, and that's true. Both donor and recipient cell do survive conjugation. Conjugation always requires a pilus. Well, the transfer of genetic material through a pilus is the definition of conjugation, so that's true as well. After normal conjugation, the recipient cell switches from F- to F+. During normal conjugation, what's being transferred is the F plasmid itself. So the recipient cell ends up with an F plasmid and is now an F+ cell. It has the F plasmid. So those three things appear to be true. What about the fourth? Conjugation cannot transfer genomic material. Well, let's take a look right here on the last slide of the homologous, or the horizontal gene transfer lecture, and you can see the F plasmid can combine with the host cell chromosome through non-homologous recombination. And when it hops back out, sometimes it can grab a bit of genomic DNA or bacterial DNA with it, and then go through normal conjugation, and it's going to take a little bit of that bacterial DNA with it. So what we're seeing here is the transfer of genomic DNA, a small piece of genomic DNA, by conjugation with an F prime plasmid. An F prime plasmid is an F plasmid that contains a little bit of genomic information. So this statement that conjugation cannot transfer genomic material, that is false. So let's mark that one. So, which of the following statements about endospores is true? Endospores are of concern to food processing because they are often found in eggs and uncooked chicken products. You might have heard of some things that are of concern to food processing because they're in eggs and uncooked chicken. That's Salmonella, which is not an endospore former. When we talked about endospores, we noted that endospores are of a danger, of a concern, because they are not killed by standard, food processing and preservation techniques. Generally speaking, the cooking or canning process is going to kill most non-endospore bacteria. But it won't kill endospores because endospores are simply tough as nails. So that's false. I mean, it's partially true. Endospores are of concern to food processing, but there's reason why that's an incorrect reason. Cells in the endospore state never reproduce. Well, let's take a look here at endospores. Let's see, where are some endospores? Here we are. So, we noted that an endospore is not a vegetative cell. And a vegetative, you think vegetation, green, growing things. A vegetative cell is an actively growing and reproducing cell. And endospores are dormant. They're non-vegetative. So this statement that cells in the endospore state never reproduce is true. They're non-vegetative cells, so they're not reproducing. Let's go on and take a look at the other answers as well. So all gram-positive cells are capable of forming endospores. That's not true, and there are lots of gram-positives that don't form endospores. Many cells come together to form a single endospore, so that was something that we talked about towards the end of Wednesday, but that wasn't in relation to endospores, that was in relation to Mycoplasma down here at the end. So this is a Mycoplasma, not an endospore. So, how would Mycoplasma genitalium stain on a gram stain test? This one was a little bit tricky because you had to use some logic or be paying a lot of attention in class. But if you take a look back at the lecture and find out Mycoplasma, so mycoplasmas lack cell walls, and you should be familiar with the fact that a gram stain is going to stain cell walls. And so gram-positive bacteria stain purple because they have thick cell walls that hold in the purple dye. Gram-negative cells stain pink because the purple stain gets washed out of them, and the pink counter stain, which binds to their membranes, is going to be all that remains. Now, mycoplasmas don't have cell walls, so even though they're gram-positive bacteria, they're not going to hold on to the purple stain. But they still have cell membranes because they are living organisms, and cells have cell membranes. So they will stain pink, so they will look gram-negative. So it would stain gram-negative. Question 6. Proteobacteria, which are facultative anaerobes commonly found in mammalian intestines, such as E. coli, are members of the blank. And this is just a matter of looking it up. And we're going to go look over here. Go find proteobacteria on this sheet. So, proteobacteria starts up here with the alpha proteobacteria. And it says, facultative anaerobes commonly found in mammalian intestines, such as E. coli. So, These doesn't say anything about facultative anaerobes down here. Glycolytic facultative anaerobes, this is the first one that is mentioned, and you'll see Enterobacteriaceae, which is the family of bacteria that live in your intestines, and there's E. coli noted right there. So it says such as E. coli, and you look up here what this is in. This is gram-negative proteobacteria. This is gamma proteobacteria. So this is a gamma proteobacteria. You just have to know the lectures pretty well for that one and look it up. So which of the following is true about viruses but not about cells? So this goes back to our, we had a table here comparing and contrasting the differences between viruses and cells. So it says, which of the following is true about viruses but not about cells? How about they may not have a lipid envelope? Do viruses have to have a lipid envelope? No. Some of them do have a lipid envelope, and those are called enveloped viruses, but they don't all have a lipid envelope. So it says some have an envelope around the capsid, but not all. All cells have a lipid membrane. And so cells, there are no cells that don't have a lipid envelope. They're all surrounded by lipids all the time. That's the thing. That's what makes them cells. They can have double-stranded DNA genomes. So viruses can have double-stranded DNA genomes. What about cells? Yeah, actually, all cells have double-stranded DNA genomes. You do. I do. Bacteria do. They all do. They reproduce by division. Cells reproduce by division. Bacteria reproduce by division. Do viruses reproduce by division? No. So this is something that's true about cells, but not about viruses. And the last point here, they may not use proteins. All cells use proteins. It's just a basic part of being a cell. It's also a basic part of being a virus. All viruses use proteins, those capsids, which were on our list of things that all viruses have to have as well. So this is not true about viruses or cells. So question 8. Which of the following is a virion? And one of the answers didn't come through here. I don't know. I think that actually it ended up that was somehow got moved to the feedback section, but that wasn't the correct answer anyways. So, which of the following is a virion? So, a single virus with a capsid fully assembled, a virus integrated into its host's genome, a bacterial virus, or I think this was supposed to say a small virus. And of them, a virion is a virus particle, which is a virus in its, metabolically inert, mostly extracellular form, where it has its capsid and, it has its lipid envelope, if it has a lipid envelope, and it's ready to go. A virus integrated into its host's genome is a provirus, and a bacterial virus is a bacteriophage. There's not really any term for a small virus. So, during which stage of the lytic cycle is the bacteriophage in a virus particle form for the whole stage? Well, let's take a look at the lytic cycle here and see whoa, okay. Sorry. I don't know if you guys heard that or not, but it played my recording. So let's take a look here at the lytic cycle overview. Sorry about that. So during step 1, attachment. Is it in a virus particle form for the whole time? Well, yes. It starts out, it's a virus particle, and it's just floating around, and it attaches. So during that whole time, it's basically outside the cell, so it's extracellular. It's got its capsid on, you see, and the genome's inside of the capsid, so beginning and end, it's a virus particle. Penetration or entry. Well, at the beginning of entry, it's a virus particle, but by the end of entry, the viral genome has entered into the cell. And so now the virus is inside the cell, but its capsid is outside the cell. So it's no longer in a virion or virus particle form. During synthesis and replication, it's making new copies of the genome, new copies of the capsid proteins, but they're not assembled together. So it's not in a virus particle. During assembly, at the beginning of assembly, it's unassembled, so it's not a virus particle, but at the end of assembly, it is assembled, so it is a virus particle. It's got its capsid on, and it's got everything in it. So during assembly, it's not in a virus particle form for the whole time, but it does end up in a virus particle form. And during release, well, it's going to start off release in a virus particle form inside the cell, and it's going to kill the cell, burst its way out, and then be in a virus particle form outside of the cell. So the correct answer here is going to be attachment and release. Not penetration, because at the end of penetration, it's no longer a virus particle. So, last question. During generalized transduction, a mistake in the viral life cycle allows bacterial DNA to enter the virus particle. During which stage does this mistake occur? Well, is it happening during synthesis, attachment, release, penetration, or assembly? Well, if you recall, during generalized transduction, the host cell's genome gets chopped up, and in some of the viruses, accidentally, the host cell DNA is put inside of the virus shell, the capsid, instead of the viral DNA. So, where is the mistake occurring? Well, the mistake is occurring during the part where you're putting the DNA inside of the capsid. And what step does that happen in, where you're actually putting the final virus together? That happens during assembly. So, those are the answers and sort of how I would go about solving them for this test. And I will see about posting this. All right. I'll see you guys later on.